Okay, then as a next point on the properties of the Bolt signal, something that is quite important for the basic analysis type that we will discuss in the winter term in statistical methods, namely the question of whether the Bolt signal is linear in essentially the stimulus properties. And this is commonly assumed by methods like the general linear model analysis of fMRI in the process of stimulus onset convolution, as we will discuss in some length in like 3 weeks or so. And it's kind of experimentally validated and justified. So the basic question here is whether this entire system of the brain and the MR signal is linear and time invariant as a function of the stimulus. So most of this happens with visual stimulation, as you will see in the following. And here is an example. So there is some visual stimulus of a certain contrast and for a certain time, this will lead to more or less neural activation. And then the question is, if this neural activation is now mapped into if this neural activation is now mapped into a neurovascular response, so the hemodynamic response, whether one from the observed fMRI signal and its expression in response to multiple stimuli can go back and infer what the initial stimulus was. So this the notion of Bolt signals, a linear time invariant system, is kind of a research topic in the 90s, and this is also then reviewed in this 20 years of fMRI article collection in Neuroimage, where this picture is from. So what is meant by these linearity properties? And these linearity properties are the properties of linear systems. So has anyone done a course on linear systems? Do they come up in physics? It's a typical engineering thing. So linear time invariant system is a typical engineering thing. From a mathematical perspective, linear systems are linear functions, and hence they have the same properties as linear functions have. The difference in terms of talking about general functions and linear systems is that these linear systems are usually thought of as functions applied to time series. But we will go into the details of that when we discuss stimulus onset convolution during fMRI data analysis. For today, we focus or we contend with some intuitive notion. So a system is called linear if its response to a multiple of its input corresponds to the multiple of the input to the basic input. So this is an example. If this is a stimulus and you get this response, and now you double the stimulus, for example, in its intensity, then you get double the response. Yeah? So basically here you, in the second thing, you have 2 times x, and the response that you get is 2 times the response that you get to 1x. Yeah? So we will make this, in terms of these pictures, more explicit when we do statistical methods. The other aspect in terms of linear functions and hence also linear systems is the additivity property. So if you add 2 inputs, so here x prime and x2 prime, so just different x's, then in a linear system the response that you get is the sum of the response to one of the inputs and the response to the other input. So from a bold perspective and simulation perspective. If you have this stimulus here, you get this response. If you have this stimulus here, you get this response. Now, if you have both these stimuli, the simplest scenario that you can think of is that you add the two responses to the individual ones, and you would observe this. So, of course, this is mathematical modeling and is mathematical idealization. So the question is whether these linearity properties of the bold signal, but of course here the bold signal incorporates both the neural processing of some external stimulus and the MR imaging of the bold signal, whether this entire system experimentally shows these linearity properties. And this is essentially what in kind of an idealized way is shown here. So on the one hand, this may be the case, so that you have these linearity properties in a time invariant manner. So whenever you send a new stimulus, these things add up and you can observe that. Or it may be the case that they actually don't hold up. So here you would see some linear time variant system behavior where the response to an initial stimulus is much stronger than the response to a stimulus later in time. And then if you add them up, you get a different response. So the main question is does the bold signal behave as a linear time invariant system where things in terms of the input, multiple inputs give multiple responses, and it doesn't matter when the input comes up or not. And this is then some famous research in this regard from the mid-90s. So again, this was done with checkerboard stimuli of varying duration that were presented to human participants. And what the important thing here was that they presented this stimulus for either 12 seconds or for 6 seconds. And then they looked at the response that they observed for the 12-second stimulation, so that's this dark line here, and they shifted and added the response to two 6-second responses, and that's the lighter line here. Can you see the difference? Somewhat, not that strong. We will see a better version. I think it's a little bit clearer, maybe. Essentially what they compare is we present stimuli of different duration. If we try to construct this response to the multiple of a shorter stimulus and compare that to the response that we see in response to a long stimulus, does it look the same? And I think it's a little bit at least clearer here. So it's the same experiment. So the thick line is the response to the actual stimulus. So here you have a 6 seconds stimulus, a 12-second stimulus, and a 24-second stimulus. You see as the thick line the actual response. And in addition, you see the prediction from shifted in summed responses. So if you, for example, think about the here predicting a 6, 12 or 24 second response from 3 second stimulus responses, then things look like this. So what do we see? Well, we see that if we try to recreate a response to 6 second pulse from the response to 2 3-second pulses, that works quite okay. If we do the same thing for a 12-second stimulus from 2 6-second stimuli, it also works quite well. Things get a little bit trickier if we try to predict a very long response. So here to a 24-second stimulus from 8 3-second responses. Yeah. So essentially, these findings in this case, in terms of the additivity and mean that it works if you don't, if you are happy with at a certain level and you don't try to go to the extreme. So if you try to predict the response to a very long stimulus using linearity from responses to very short stimuli, there might be some differences, but if you're roughly in the same ballpark, then it works relatively okayish. So you can also see here, so trying to predict a 24-second response from two 12-second responses seems to work okay. This is another response, another finding in this regard. So what they did here in a different experiment by Dell and Buckner is that they presented visual flashes, not checkerboards, to participants with a 2-second interstimulus interval, and based on the response to 1 stimulus, 2 stimuli, and 3 stimuli, they estimated the response to the first, the second, and the third stimuli. And the question is here whether these responses are roughly the same, so whether things are time invariant, and they roughly are, although there's a little bit of a decrease for the second and third stimuli as compared to the first stimulus. So there seems to be, also not very surprisingly, some habituation or adaptation. And this is then looked at in more detail here, namely as a function of the interstimulus interval. So the question is, if you give stimuli closer together in time, do you still always get the full response, or is there more and more adaptation? And this is actually what is found. So here the black line refers to giving checkerboard stimulation and after basically rest, so nothing happened, and then comparing this to the response to a checkerboard when previously a checkerboard was presented just 6 seconds ago, or just 4 seconds ago, just 2 seconds ago, or just 1 second ago. And as you can see, the response to a checkerboard when just a second before, 1 second before another checkerboard was shown, is much lower than as if no checkerboard was shown for a long time. So that essentially argues for some adaptation that is specifically strong if the interstimulus interval gets quite short. But here around this time, let's say between 5 and 6 or so seconds, there is not much adaptation. And why this is interesting is, of course, the question of how far apart you want to put your trials in time, so that you observe to each trial a good response, and don't get into adaptation processes. So the bottom line of all of this is essentially that this linear time invariance holds if you are not going to the extremes. So if you put stimuli too close together in time, maybe even a little bit closer than here because it's not as extreme as shown here, then you observe time variant behavior. So you observe adaptation like in any biological system. So you don't want to do this, then it's not time invariant anymore. And in terms of summing things up, if you don't try to predict very long responses from many very short responses, or essentially this, then if you're not going to the extremes, then you have this linear time invariant behavior. So that's the story there. So if somebody asks you, is Bolt system a linear time invariant system? The answer is in a way yes, under good conditions. So not under all conditions, but if as long as things are kind of nice, then yes. And this idea that you can model the Bolt system in response to a stimulus as a linear time invariant system, this is then of course used in the analysis and the prediction of data and Bolt data when you do GLM analysis. So we come back to linear time invariant behavior in a couple of weeks. You can also already read about it in the statistical method script that you of course have. Yeah, good. So this a little bit on the linearity properties, for which there's some empirical evidence. And now jumping to another topic, namely the question of signal to noise ratios. So the question is, or what this is about, is essentially learning about different forms of signal to noise ratio and being aware that Bolt, like any biological signal, is not as beautiful as some kind of measurement of a physical constant or something, but is inherently noisy. That's the main point. So that this biological data is noisy. And one important thing first to take note there is that Bolt signal changes that are induced here, for example, by hand squeezing, are quite small if compared to the total MR signal intensity. So if the MR signal intensity is just the gray value that is encoded here, changes that you see over time are not kind of doubling the MR signal at a given area, but the typical percentage signal change is around 4% or something like this. Yeah. So if these gray values, for example, are numbers like around 100, then they would be around 100 during rest, and then if there's something happening, they would go up to 104, 105 or something like this, but not to 200 or something. Yeah? So it's subtle changes that are induced by stimuli. So this is what this also shows. So here, if you compute the percentage signal change, so instead of looking at the raw MR signal, which also doesn't really have a useful unit, it's just values around a certain baseline of like 100 or something, depending on the setup, then you see that the percentage signal change is roughly in the order of 3 to -1%. Yeah, so it's quite small. But this doesn't really matter. I mean, this is always, of course, in science. One question is how big a change is. The other question is how reliable it is. Yeah? And this is the kind of whole tension system that you deal with in science. So if you have something that is really reliable, highly significant, then it might be meaningful. But if it's only very reliable, or if it's that goes into the question of how reliability is measured. But if it's very reliable but very small, then it might also not have any meaning. On the other hand, if it's very reliable and small, it might also have meaning. So it's just it's this tension system where science basically happens. Now, if people talk about signal to noise ratios in fMRI, they have different things in mind. And of course, whenever you hear that, but the signal to noise ratio is really low, the question is, of course, what is the signal and what is noise? And the thing is, with signal to noise ratios, like many things in science, it's of course a subjective claim what a signal is and what noise is. So for a certain scientist, signal is some meaningful change in some quantity, whereas noise is a non-meaningful change in some quantity. And the signal to noise ratio would be then the relative strength of the signal as compared to the noise. The important thing is that what some people call noise would be a signal for other people. So often certain aspects of signals are ignored for some time, and then somebody says, well, we should look at this part of the signal. So for an example is, for example, these trial by trial analyses. So if you average in e.g., if you do event-related averages, or in BOLD, you of course only look at the average, and any thing that is different between trials, you would say, yeah, this is noise. You can, of course, also argue differently. You can say, yeah, on a trial-by-trial basis, the cognitive state of the participant changes. So we should look at the trial-by-trial covariation of some cognitive variables with the observed signal. And then what the people that average call noise and unimportant variability would be actually important variability for you. So yeah, to say this has really low signal to noise ratio is a little bit meaningless. One should always, when talking about signal to noise, be a little bit more specific in terms of the signal to noise ratio that one is interested in. And there are like three or four, no, I think it's 3 different signal to noise ratios that people talk about in fMRI. One is the raw signal to noise ratio, and that's the ratio between the MRI signal intensity associated with the sample, in this case the brain, and the MR signal intensity that is measured outside in the sample, so outside the brain. So this essentially just measures, are the gray values that we see inside the brain, are they different from the black values that we see outside the brain? And of course, if you can't see a brain at all on an image, although you're trying to image that, then of course that means that something is going wrong. So this raw signal to noise ratio is just a measure of the scanner hardware. So if the scanner is broken, then you might have a very low raw signal to noise ratio. But what's actually happening is that, of course, also the technicians at the imaging center, they would constantly measure phantoms and compare the signals that you get from phantoms. So essentially just balls or fluid-filled things like bottles. They would compare the signals from these water-filled bottles to what is going on outside and make sure that the raw signal to noise ratio is okay. If it's not okay, and there's no sense in doing any experiments. There is something called contrast to noise ratio, which is a little bit a confusing term, because if you hear contrast, then of course you think more in terms of statistical contrast, but this is not meant here. So the contrast to noise ratio in MRI refers to the magnitude of the intensity difference between different image regions divided by their variability. So this is a measure of the detectability of tissue differences. So if you have low contrast to noise ratio, that means you cannot really distinguish different tissue types in your anatomical imaging. And then there's something called functional signal to noise ratio, which is the ratio between the intensity of a signal associated with changes in brain function and the variability in the data due to all other sources of noise. And this is, of course, the basic measure for the detectability of condition differences in functional neuroimaging. And what we will see in statistical methods is, of course, that this functional signal to noise ratio is exactly what is evaluated using general linear models, and then comes down in terms of a quantitative measure to T statistics or F statistics. But in our case, it will come down to T statistics and their associated P values, because that's what we focus on in statistical methods. So functional signal to noise ratio is essentially the result of your statistical analysis and refers to bold signal changes over time. So this shows an example for functional signal to noise ratio. So one way to think about this. So here you have a voxel and it's doing something, so it's going up and down. Here you can see that there was actually some stimulation. I think it's about motor stimulation, and you see that this voxel clearly, if something happens, it goes up, and if nothing happens in terms of the experimental condition, it goes down. So if you do a histogram analysis, for example, and you now label all the data points that happened in these kind of periods, you label them red, and you look at the distribution of this histogram here, and the other ones that are lower, you label them blue. Then you see that the distributions of these values, or the empirical distributions of these values here, are not that overlapping, while if you have this data, they are much more overlapping. And of course, the quantification, the basic measure of quantification for this distributional overlap will then be a t-value, where you have a low t-value if you have this kind of overlap, strong overlap, and a high t-value if you have this little overlap and good separation. Yeah. So when you read the Hüttel on the functional signal to noise ratio, it reads a little bit weird in the sense that there's not an explicit link to the typical analysis that are made, but exactly these kind of aspects of the data are of course picked up when you do statistical evaluations of the time series here. And how that works we will see at length in statistical methods. Another aspect that is also important to keep in mind is that if you hear about functional specialization and that certain brain areas show response, for example, primarily to faces, or we heard in Rainer Goebel's talk that area in T responds primarily to motion. That doesn't mean that this area doesn't respond to any other kind of visual stimulus. So if you provide visual stimuli, you usually see the entire visual system lighting up. When maybe tomorrow or on Monday I'll show you the basic analysis that we want to discuss in statistical methods all the time. That is actually the response to a visual stimulus. You will see that most of the back of the brain is actually lighting up. However, then you might have areas in the brain that, if presented with faces as compared to tools, might still show a reliably stronger response. Yeah? So that these areas that show a preferential response, they also respond to other stimuli, but they show stronger responses to specific kind of stimuli. Yeah? So that's just something to keep in mind, that you don't think the brain doesn't do anything and FFA, so the fully formed phase area, only ever shows neural activity and bold activity if there's a phase. Good. So noise sources. So I said, or we discussed now, that there is quite some noise and the signal needs to be differentiated from the noise. So what are typical noise sources in fMRI? And these noise sources, like all signals, can be characterized essentially in terms of their temporal features and their spatial features. So this focus now first on the temporal features, and what you see here is again a frequency spectrum. So you see some frequencies and you see their power in MR time series. And what can be observed here are two peaks, one on the 1.5 Hertz scale that is likely to result from cardiac pulsation, and another peak that is on a lower temporal frequency and relates to changes in CO2 in the brain that are induced simply by respiration. So for the moment, this is mainly to show you that you can look at temporal features of noise. You can also look at spatial features of noise. These then usually relate more to motion and are mainly expressed well in the eyes, but that's something where you don't have much of a problem. The bigger problem is that you have changes in the MR signal due to the fact that people move different parts of their brain in and out of a given voxel's imaging area, and these kind of things then mainly show up at the border of the brain, because there you have these strong tissue differences. So this was mainly just to characterize, or to be able to say that there are temporal features of noise, there are spatial features of noise, but what are now the noise sources? And there are a couple of noise sources that always play a role. So the first thing is thermal noise, which is just noise that results from the thermal motion of electrodes in the scanner hardware, and there's actually nothing that you can do about it. So your MR signal intensity fluctuates a little bit due to the scanner setup. So it's the same thing as if you turn on a speaker. You also hear, if you amplify a lot, you hear this white noise, and that's of course present in all kind of physical electrical systems. Then you can have something that you might refer as system noise. That's a fluctuation of the MR signal induced by the scanner not working properly. This is of course something you want to take care of, and of course you also don't want when you do an imaging session. Even if the scanner works well, what you always have in MR signals is scanner drift, due to a slow fluctuation of the static field over time. So remember, there's this B0 field, it's supposed to be constant, but if you actually do an imaging session, the entire field slowly varies over your experiment, meaning that also your MR signal slowly varies over the experiment. But of course, as we will see in a second, this can be easily taken care of. The biggest problem in terms of noise is physiological noise, because that is, first of all, not that well predictable, like for example scanner drift, and especially if it's not uniformly distributed over your conditions, can of course induce artifactual correlations with stimuli. So for example, if you have one condition where people always nod their head because just because they do so, and in the other condition they don't, then of course if you try to tease apart the signal in response to the two conditions, one of them would be polluted essentially by this motion artifact. So physiological noise is an important thing, and of course the aim is to minimize this. Scanner drift, these low frequency changes can just be filtered out, like we discussed for the EEG. This is low frequency activity that is on a very much lower than your signals in terms of its frequency. It's your signals of interest. So just by applying a high pass filter, you get rid of that. But you will read about it if you do fMRI pre-processing. At one point there will be some high pass filtering to get rid of the scanner drift, and then you know what this is about. So in addition to, this is not really physiological noise, this is more like physiological variability in different brain areas and in different subjects. You of course also have variability like for EEG that you can consider as noise, or you can consider as a signal. So for example, different both signals in different brain areas of course often can be an interesting signal in terms of functional specialization. Other thing in terms of one in terms of variability from participant to participant. This is of course now something that people often report and are interested in terms of individual differences. So yeah. And then it becomes interesting. If you say all humans are the same and you want to describe what a typical human response is to a certain stimulus in the brain area, then of course it's noise if different participants show different responses, like shown here. But if you say, yeah, people are different, so they show different responses, and you find some other response where people actually correlate with this, then it's of course very interesting. And you can say something like, people who have a stronger response in this brain area are better at doing this and this. This means that this brain area does something. So it's always a question of what you're interested in. Finally, also data analysis can create the impression that there's quite some variability. So here, this is from the Hüttel, you can get the impression that visual cortical activity is quite different between participants, but the point here is so that you sometimes don't have any visual activity and sometimes you have visual activity. The point here is that this is kind of artifactually introduced by thresholding the images, and if you go to a lower threshold, you will again find that the responses are fairly similar, although expressed at different scales. So yeah, so this is basically arguing against looking at or threshold. So basically against arguing against using thresholds and then claiming that responses are all or nothing. Usually they are there, but in kind of graded or scaled to a scaled degree. Good. So the last thing, with my last energy for today, I will do are these field strength effects, and then we can relax. So these field strength effects is basically the question. We have a 3 Tesla scanner here. Why do we have a 9 Tesla scanner? Wouldn't it all be much more awesome if we had a 9 Tesla scanner? And the answer is yes and no. So the raw MR signal increases proportionality to the field strength, and in its square. So if you double the field strength, you get 4 times the MR signal, so you get more signal. And the thermal noise doesn't increase quadratically in field strength. So one would, if one just cares about thermal noise and the raw MR signal, one would actually be interested in increasing the field strength. So ideally, this would be that mean that using higher scanners helps, and it definitely helps for the raw SNR. But of course, what we are interested in BOLD is not so much the raw SNR. Remember that the raw SNR was the difference between sample voxels and non-sample voxels. So basically the difference between the black part outside the brain and the gray values inside the brain. But what we are interested in is the functional SNR, so whether dynamic changes are reflected in the MR signal. And there, the problem is that although the MR signal so this is field strength and this is the relative intensity, the MR signal is going up quadratically in the field strength, and the thermal variability is only linear increasing, meaning that you have this increase in raw SNR. Also, the effect of physiological artifacts increases superlinearly, meaning that the functional SNR doesn't increase as much as you would hope from just thinking about the raw SNR. Question is, of course, I deleted this slide. The question is, of course, why this is, and this is then again like a more an active field of research. The finding is that if you have a higher Tesla scanner, and you do phantom imaging and so on, the signals are much better. If you then do fMRI studies, you find that your T values and the signal differences between different conditions don't suddenly get a lot better. So then the question is, what does this relate to? And the explanation is that also the noise contributions increase. So from this and kind of this leveling off of the functional signal to noise ratio, what you care about in fMRI, the bottom line is that it not necessarily as a general purpose tool helps to go to higher and higher field strength. At least that's kind of what the general story is. And then, of course, there are other things. So, for example, if you have a higher 7 Tesla scanner, you can go to a higher spatial resolution, like Rainer Göbel showed for MT. But then this might only you might only actually image visual cortex, so you only focus on certain aspects. Another thing is that if you go to higher field strength, the dangers of imaging increase, of course, and also how well participants tolerate these stronger field strengths actually varies quite a lot, because they can induce, so if you move in a very strong magnetic field, this of course induces electrical activity, and you get these phosphene phenomena, so that you see flashing lights although there's nothing flashing, and this is of course also not nice. So there are other disadvantages to having higher field strengths. So this 3 Tesla scanner model that is mainly used for fMRI kind of is doing okay on the functional SNR, and it doesn't have the strong invasive effects and necessarily additional cautionary constraints as has a higher Tesla scanner. So now my energy is 2% before 0, and if I would go on, I would collapse here. So that's the thing that we discussed. So then tomorrow we're going more cognitive and then also more data analysis. So any questions right now?
I'm wondering, while reporting their results, how often do scientists report their non-meaningful data in this context, their noises they have?
Well, tricky question. So if because the question is not quite clear. So in a T value, if you have a T value, then of course a T value is always a ratio between the effect size and the noise. So in a way, when reporting a T value and a statistical map, you also report your noise. If the other question is, of course, if you don't find anything and you would, based on your framework, call everything noise, so you have a complete null finding, then of course it's a good question, because usually null findings are not published, which is a problem for the entire scientific literature. So if, for example, these people here that just had this Nature and Neuroscience thing, if they wouldn't have found these differences between areas, if they wouldn't have, where are we? No, this is this. So if they would for both Bold and Vaso wouldn't have observed these differences here, they wouldn't have published. So it's not so much them that they wouldn't have reported it. They would like to have reported it, but Nature Neuroscience wouldn't have said, well, that's awesome. There are no differences between superficial and deep layers. Let's publish that. So that wouldn't happen. So this is actually a big problem in terms of that usually only results that show some signal actually are seen in the literature, whereas pure noise results and hypotheses that cannot be rejected and so on don't show up. And that's more a problem. From my perspective, that's not necessarily the scientists that are the problem there. I mean, there are definitely certain scientists who have this very much internalized, that this is important to have something significant, but it also has to do with kind of the journal culture, and what I think it has a lot to do with what people think is interesting. And although most people would say, well, it's also important to report null results, then there's some self-censorship in a lot of scientists that say, no, I cannot write a paper purely on null results. But it's important, and hence we are all, or at least me, active in this open science movement, which actually tries to push things into a more balanced view of things, and actually just a more realistic view, and not focusing so much on awesome results and so on. So if you're interested in open science, by the way, we have an open science working group at FU, and we're meeting today again at 5 in the library. If you're interested and you know what open science is and why it's important, feel free to join. I will send around that to the scan email list in a second, and you can see it. It's sometimes tricky, so we had a course on open science and do stuff on open science. It's sometimes not that easy to get young people into open science because they don't know how fucked the system is. You first need to know, you need first-hand experience about the fuckness of the system until you want to change it. If you come kind of naively, like when I went into the system, thinking that it's science, it's academia, everybody's very honest, and everybody's interested in finding the truth or something, and they don't do it for their career. That's not the case. But you need first to experience that so that you can appreciate that something needs to be done and we need to change the system. With this, then I see you tomorrow. Or maybe this afternoon I will send this email around. Then you know where to go if you have time and interest. Goodbye.